Hi. Thanks for coming. How many folks I'm Jamie Pearson. I'm at the Horn Point Lab, part of the University of Maryland Center for Environmental Science, and I am an oceanographer, biological oceanographer, and I study plankton ecology. How many folks were here last week? I recognize. Okay, great, perfect. Okay, so last week you heard from Mike Roman, and he talked a bit about how hypoxia forms, what it looks like in the ocean globally, in the bay, and all that kind of stuff. I'm going to talk a little bit more today about what it means for the animals that are trying to make a living there. And I'm going to focus on animals. I'm going to focus on zooplankton. Everybody know what the plankton are? Pretty much?
We took your classes.
Right, well that was about plastics and plankton. But plankton are all the organisms that drift around in the ocean. So they can be, plankton can swim, a lot of them, not all of them, a lot of them can swim. They can be all different sizes, but they can't, what makes them plankton is they can't swim stronger than the currents in the ocean. Okay, so fish can swim against the current. Fish swim upstream. If you think of salmon, if you think of striped bass here, they can swim. Plankton cannot swim against the current, and that's what makes them. So we're going to be talking about that, and we'll be talking a bit about fish and jellyfish and things like that today. So first,
They drift.
they drift. That's what the word plankton means. It comes from a Greek word that means wanderer or drifter. Okay, so before I get started, and yeah, please ask questions as I go along. If you look at me like I'm crazy and it seems like you're not understanding, I will try to fix it, but you can also just yell at me. Or yell to me.
Is that really true? Is that Cranberry?
Hightstown. Yeah, my grandmother lived in Cranberry. Yeah, my grandfather sold his farm to the Turnpike in '63 for Exit 8A, when they put Exit 8A in Forest Gate. So,
Wow.
so I was born and raised in New Jersey. I got my biology degree at the University of New Hampshire, where Mike did his PhD. I worked as a technician for a number of years at the University of Rhode Island, where I started going out to sea. I spent about 3 or 4 months of the year on large boats doing work on Georges Bank, studying plankton, the link between plankton and fish and climate change and things like that for a project called Glowback. I decided after a few years of that, I decided I really liked this business, but I wanted to be the one asking the questions. And so I moved out to Seattle and went to graduate school, where I got a master's and a PhD at the University of Washington in ocean, and my degrees were both in oceanography. When I finished there, I came to Horn Point, part of the University of Maryland Center for Environmental Sciences, to work with Mike Roman, working on a project. I started out working on a project in the Upper Bay. Mike also gave me what he called a honey license. He let me write proposals and bring in some money, and I actually showed up with a little bit of money from a project that I had written with my advisor at University of Washington. And when you start bringing money in, they let you stay. And so I have been at Horn Point since about 2006. I've had a number of positions there. I started out as a postdoc. I was promoted to research faculty, then tenure track, and now I am a tenured associate professor at Horn Point. And like I said, most of my work has been in zooplankton. I'm doing a little bit of work with microplastics these days, and I do some work with folks in Puerto Rico, sort of broadening participation, enhancing opportunities for students to get involved in ocean science. So that's a little bit about me. Been on the Eastern Shore since 2006. So before we dig into the animals, I just want to do a real quick overview. Mike showed this slide, and I really like it because it talks about how much, this diagram here talks about how much oxygen water can hold, and how it's affected by salinity here on the x-axis and by temperature on the y-axis. The colder and fresher, the more oxygen the water can hold. So when it's colder and fresher, it can hold more oxygen, the solubility is higher. When it's warmer and saltier, it can't hold as much. That's the blue colors over there. And these boxes represent some places in the world where there's where we have a lot of low oxygen water, the Chesapeake Bay and the Gulf of Mexico. And so if you look at the Chesapeake Bay, for example, the solubility, the amount of oxygen at 100% saturation ranges from between about 9 to about 7. So that's a pretty big difference over the range of temperatures that the Chesapeake, this is temperatures and salinities that the Chesapeake experiences in the summer when we have low oxygen here, okay? So the things to remember are there's only really two ways to get oxygen into the water. Photosynthesis, that has to happen in the surface with light. That's all the algae in the surface, the phytoplankton. They're photosynthesizing and they're producing oxygen, or mixing at the surface, right? So wind mixing, creating bubbles by the wind right at the surface layer. So you only put oxygen in the water right at the surface. But we use up oxygen in the water. Organisms use up oxygen in water down deep, okay? Everywhere, everywhere. So photosynthesis and mixing are happening at the surface. But everywhere, organisms are respiring. That's how they get their energy. They're using up the oxygen in the water.
Does that mean that algae is good? If they're producing.
Well, algae is just algae. Whether too much of anything is I think Mike said this in his talk, too much of anything is a bad thing, right? So yes, algae produce about half of the oxygen in the atmosphere that we see. Trees and grasses. If you think about it, the ocean is covered, 70% of the surface layer of the Earth is covered in ocean. And there's algae in pretty much all of that, and they're producing oxygen. When it gets bad is when we put too many nutrients in and we stimulate more production of algae, and then they die and they sink out, and that's when we get low oxygen, because we're respiring it. We're all that material, all that algae sinking into the bottom is being respired and used up without being able to put new oxygen in, because that only happens at the surface. Okay? So, before we go too much further, I'm going to talk about a couple of different things. I just wanted to get a couple of definitions. I'm going to say OMZs or ODZs. These are the areas in the ocean with low oxygen that Mike talked about. So I apologize if I use these interchangeably. I just wanted to mention it. Some of the slides, or I might say DO, and that just means dissolved oxygen, concentration of oxygen in the water. We talk about hypoxia and deoxygenation. These are kind of synonyms. This means low. I'll use both of these for low oxygen. And usually the agencies define low oxygen as like less than 2 milligrams per liter in terms of concentration, or using these other metrics. Chemists like to use micromole. I'm not a chemist, and I don't play one on TV. But I wanted to. The first thing I wanted to do before we get into this is talk a little. I mentioned here that what uses oxygen in water is respiration, and this is. I'm gonna go through something really quickly in a couple of slides. Because this is something that really struck me when I was in graduate school and I had to take a marine chemistry class. And it's a way of sort of understanding the big picture about respiration and oxygen in the ocean and the circulation. It kind of ties in the global circulation and the oxygen and respiration. So has anybody heard of the thermohaline circulation or the global conveyor belt? Mike mentioned it briefly, global conveyor belt, right? Okay. This is a map that is sort of an idealized view of the conveyor belt of the ocean. And this is something, Mike, did Wally Broker come up with this? He's credited with it.
He worked on it a lot, yeah.
Yeah. So this is the general pattern for how water moves around the ocean on a global scale. And there's a couple important points here. There's only two places on Earth where we form deep water. In the North Atlantic here, and down here off of Antarctica. And what happens here is as this warm water is moving through the tropics, it's evaporating and it's getting saltier, and then as it moves north, it's cooling off. So when you have water that is salty and cold, it gets very dense, and then it can sink down deep. And then when it sinks down deep, it travels around, and all this is driven by gravity and by moving by water sinking and water rising and things like that. So it moves all the way around, and it comes up in a couple of places. It comes up here and it comes up here off of the Horn of Africa and in the Indian Sea, and it comes up in the North Pacific. Okay, this is generalized. This is a very general position. But if we know that deep water forms here, and we know that there's this big global pattern. Oops, wrong one. Okay. Oceanographers can use this thing called apparent oxygen utilization, And we can use it to estimate how long it takes water to travel around the globe. And this is going to relate to all of the stuff about animals in America. So anybody, those are a couple folks that said they've seen this or heard this. Anybody know about how long, or anybody want to guess how long it takes water? If you form deep water up here off of Norway, how long does it take before it comes up again in the Pacific? Any ideas?
A year.
A year? That's a guess. Yeah, we like a year. All right. So if we have a little bit of information about the oxygen in the water, we can measure oxygen. We can measure the amount of oxygen in the water, and we can estimate the solubility of oxygen. How much should be there when it's fully saturated, when the water is fully saturated. And if we remember that, the only place we put oxygen in the water is at the surface, right? So only here in the North Atlantic and off of Antarctica. And then it travels around, and we're using up oxygen the whole time, okay? And this is what the dissolved oxygen concentration looks like along this line. And this would be the south, so if you turn this sideways, so we have very low oxygen here in the North Pacific, which is what I kind of mentioned over here, right? So this is really old water here in the North Pacific. So we have really low oxygen, and we know what the solubility should be. We can calculate how much was used up on that.
I'm changing my guess.
What's your guess now?
I'm guessing longer because the cold, it's coming from the depth and it's probably not moving.
So what is your guess? I mean, longer than a year could be.
I'm going to be 20.
20 years? Okay, a decade, 20 years. Okay, great. So this is what a map of oxygen utilization looks like all over the world. So can we use it to predict how long? So don't worry too much about the numbers here, but oxygen utilization is about 300 micromoles per kilogram of seawater. The respiration in the deep ocean is very, very slow. It's cold. Average temperature in the deep ocean is about 4 degrees Celsius, okay, for a whole bunch of reasons. And the density of seawater is about 1,000 kilograms per cubic meter. Come on, here we go. So we can do a pretty simple calculation, and we wind up with about 300, yeah, don't worry about the math, I did the math for you, okay? Well, actually, John Hedges, my marine chemistry professor, did the math for me.
840.
It ends up being about 842 years, okay? So this is one of the things that I love about oceanography is that we're connecting the biology, the respiration, we're connecting the physics of the moving of water, and we're connecting the chemistry of the solubility of oxygen in the water. So in order to understand all of this, you gotta know a little bit about all of these different things.
Okay, so I was going to ask,
Okay.
how do you start from one point? What do you do? Put it in the water, and how do you go?
So this is just an estimate based on our measurements. If you remember the map that Mike showed with all those many measurements all over the globe.
Yeah.
And if you run, we've done a lot of model simulations. The global alien circulation was postulated at least as early as the 60s or 70s, based on global maps of what the temperature and the salinity and the overall surface currents of the ocean were being. We knew that if the currents were moving, water had, the ocean's not getting well, okay. With sea level rise, the ocean is getting deeper, fine. But in general, it's not, it's essentially at equilibrium. And so if water's moving from one place to another, it has to be moving, it has to all balance out, right? We need a mass balance. And so by doing that, oceanographers put together this really pretty map of the global alien circulation. And then from that, figured out using these fairly simple calculations with just a few measurements about how long it takes to do that. So we estimate, we fudge a lot of things sometimes when we're just talking. So 800 to 1,000 years. These are estimates here. So it takes 800 to 1,000 years for water that sinks in the North Atlantic to come up again in the North Pacific.
Did any of you see that movie Day After Tomorrow?
Yeah.
So, how the weather changed. Also, this affects the climate a lot, this deep water sinking. So as we warm the oceans and all of that ice in Greenland, things are melting, that's coming in the North Atlantic. And what Jamie said is the water gets saltier, but if you have fresh water coming in, it's gonna mix with it and not make it sink, right? And so if you shut off that thermal healing circulation, it's gonna change our weather pattern.
Well, glaciers are fresh water, they're made from,
Yeah, glaciers are on land,
right, so
right, so they're,
just
yeah.
made from what comes.
Yeah.
From the sky.
Yeah, so there's a lot, I mean, I could do all day on this. We're going to get to this, but this is, to me, this is fascinating, and I wanted to mention that part because it connects to the respiration, that use of oxygen by the organisms that are in the water, right? This pattern, this global thermohaline circulation, thermo is heat and saline is salt. So this pattern moves salt and heat around the globe, and it actually, this pattern, as Mike was saying, it regulates climate. So we see changes in this, we see changes in climate. Carries, the ocean, water holds a lot of heat, right? It takes a lot of energy to heat up or cool down water, and so this moving around is actually carrying heat with it throughout the ocean.
So it seems like almost every year, the meteorologists come out with, oh, it's gonna be an El Nino year, or the opposite based on the currents. So how does that happen so
And how does that affect this?
yeah, if this is takes so much time, how is that?
Come to my Chesapeake Forum class next year, when I study up to answer that question. I'm a plankton ecologist, remember? You've hit the limit of what I know. So El Nino is a whole different thing. That works at a shorter cycle.
That's a whole class.
Yeah, there's a whole class on that. I'd love to talk about it.
They are increasing them, we're having more
Yeah.
algae.
Yeah.
Right. But the thing I wanted to just sort of, I wanted to show how the global circulation and how oxygen utilization by organisms is all really connected here. Okay, and how we can use it to learn things, how we can use it to learn about these patterns.
Is diffusion a factor in this?
It's really.
Does it really depend on?
Yeah.
All right, so I'm going to explain that.
Yeah. So now we know that respiration happens in the ocean, that it affects the operation, how the ocean moves around at global scale. We can measure it, we can do all. But what we came here today to talk about, 20 minutes ago, when I started, is animals, right? And the organisms in the water and how they deal with low oxygen. And so how does oxygen get into organisms? It gets into organisms through diffusion. Diffusion is an inherently very slow process. And so if you think about the global ocean, anybody know, I'm doing my basic oceanography here. Anybody know the average depth of the ocean? I told you that the deep water is about 4 degrees Celsius. Average depth of the ocean, any guesses? 20 miles, it's less than that. Yes.
You're going big.
All right, in the interest of time, so we can talk about animals here. It's about 4 kilometers deep, okay? So diffusion is a really slow process. So diffusion doesn't really, the way the currents move is much faster. So moving oxygen around in the ocean by the currents is much faster than through diffusion. But at small scales, diffusion is how oxygen gets into organisms. Okay, so this is the outside of the organism. This is the inside of the organism. And so when we have higher concentrations or higher partial pressures of oxygen out here and lower here, the oxygen molecules will move across that membrane, across that layer. Right. So the way that oxygen gets into organisms is through diffusion. Now that can happen in lots of different places. And because I'm talking so much, I'm going to skip through a couple things here. Animals have evolved a variety of structural forms to make that diffusion more effective. So we use lungs, right? Lots of animals on land use lungs. Things like amphibians can use external gills. Things like other invertebrates can use internal gills. And then some other arthropods and things use trachea. A lot of the zooplankton, the ones that we study, they're small enough the organisms are tiny enough that they just, oxygen diffuses across their outer skin. They often have pores, if they have a shell, they have pores in their shell, and so a lot of them have just oxygen diffuses across their outer skin. That's how they get oxygen into their body. And then copepods, the things that I study and that Mike studies a lot, do not have blood, but they use other ways of moving the oxygen around to get to all of their cells. But they're really small, so it doesn't have to move far compared to a whale, right? Or compared to a tuna or something like that. So tuna has gills and a circulatory system, and that's how they move oxygen around to all of their organs and things. With really small animals, some of them just absorb it through their skin. Okay?
Are zooplankton multicellular?
Like,
So, yes and no. So some are and some aren't. So zooplankton is a lifestyle. Zooplankton are animal plankton, things that eat other things for their energy. So they're single celled
Okay.
and they're multicellular. So most of what I work on are multicellular. So this is all well and good, but we're talking about low oxygen, right? So they have a variety of structural forms. Some get oxygen through special organs, and so how do they manage when they have low, when we're in low oxygen? They can adapt by adjusting those structures. So it's all about surface area, right? The more surface area, the more oxygen you can get into those structures. That's why they're all very like dendritic, right? They have lots of little, because that increases the amount of surface area that's in contact with the water where the oxygen is. So the more surface area, the more space for them to come in. They can increase the chemical efficiency of the things that bind the oxygen in the blood. So we use hemoglobin. Other animals use lots of different things, and I'm not a physiologist, so don't ask me what they are. But there are lots of different other chemical enzymes and things to grab onto oxygen, right? Or in some cases they can move. Now, if you're an oyster and you're stuck on the bottom, you're kind of screwed if the oxygen goes low. You can't really move. But if you're a fish or lots of plankton or things, you can move out of the area. You can move up. If you're a fish, you can move sideways. So that's one of the things that lots of animals do to deal with low oxygen. So this is just sort of going over this. Mechanisms to efficiently remove oxygen from water. They can increase the mechanisms to remove the oxygen from water. So that's by making more surface area, right? Or by increasing the types of, by changing the types of enzymes that they use to grab the oxygen and transport it. They can reduce their metabolic rates, basically slow down breathing so they need less oxygen. That's something else that they can do. And they can undergo anaerobic metabolisms. They can use other chemicals to get energy, right? So the reason that they need oxygen is oxygen is used. It's broken apart, and the energy stored in the oxygen molecule is used to break down other materials. So that's how they get energy. It's by respiration and metabolism is a process by getting energy by breaking down molecules that are storing a lot of energy. So they can come up with other ways to do. I'm not really going to talk about this. This happens a lot in the microbial world. And I'm not in bacteria and things like that. I'm not really going to talk about it too much. But lots of organisms, if you go for a long run, you have lactic acid buildup, right? That's an example of anaerobic metabolism where you've used up all your muscles have used up all the available oxygen, and they're switching to a different mode in order to get some energy to keep going, right? Okay. So. In some animals, so there's different ways of sort of approaching this. Some of them, there's two types that have been identified, two types of approaches, the oxyconformers and the oxyregulators. And the oxyconformers have a respiration rate that's dependent on oxygen concentration. So these organisms need, the more oxygen, the higher the respiration rate is going to be. So they're dependent on the amount of oxygen in the water for what the respiration rate looks like. And the oxyregulators usually have a much lower respiration rate, but they're not as affected by changes in oxygen. So they deal with low oxygen in a different way than these guys. Okay?
What are examples of each?
I'm glad you asked. This is a type of shrimp called a mysid. And this is a really good example. I think this is one of Brad Seibel's papers. And so in this example, this organism actually switches between oxyconforming and oxyregulating way of life, depending on its activity level, okay? So if it's swimming this is oxygen concentration on the x-axis, and this is its rate of oxygen consumption, so this is its respiration rate here. When it's really active, you can see that it increases its respiration rate with higher oxygen. As the amount of oxygen available goes up, its respiration rate goes up, okay? When it is at low activity, that should say activity, not activity. I typed that. When it's at low activity, it maintains the same respiration rate regardless of oxygen here, okay? So that's one example. Some organisms are more prone to one or the other, but this is an example of a mysid. This is about this guy is about 5 inches long or so as an adult, and it lives in the low oxygen waters, like these OMZs off in the open ocean. And it is able to switch between these modes as it migrates up and down in and out of the low oxygen water.
Or oysters, oxyregulating? They kind of have,
I should have anticipated that question.
they kind of have to be, or, well, I know they don't do well.
Well, they can't pick up and move.
So
Right,
maybe,
they can't pick up and move.
right, so they almost have to have that ability. Unfortunately, it's not enough of an ability to help them.
Yeah, I think that, well, they could do something like this, right? When they're really active, when they're actively feeding, for example, actively pumping water, they might be more oxygen conforming, and then they might go dormant. Oysters also can close off and go dormant when conditions are really bad, whether it's in the winter or whether it's in the summer.
What is it, like if it's too much salinity or?
Or low salinity usually is a bigger issue in a lot of coastal areas. If you get big runoff, or 2018 when we had record rains and things, I think they saw some of the oysters had low growth. And preface this because we're being recorded. I don't study oysters, and there's a lot of oyster experts at Horn Point. I'm happy to point you their way. They'd love to talk to you about oysters all day long. Okay.
And crabs.
Yes. So crabs are different though because they could move. So if they get stressed out, crabs are likely a little bit more. Now this is a crustacean, and crabs are crustaceans too. So perhaps they could have these two. They might have a similar multi-mode response where when they're not very active, they might be more of an oxyregulator, and when they're active, they might be oxyconforming. But they can also move. Crabs can pick up and leave, and we see that, right, Mike? The jubilees, right? So especially down like in the Gulf of Mexico, when you have in the Gulf of Mexico, there's right off the coast, there's these large dead areas that are dead zones that are driven in large part by runoff from the rivers and things. And what happens is when you get specific wind events, it'll push some of that low oxygen water up onto right up into the shallows. And you see this in other places. In Hood Canal, you see fish die offs and things like that. And in the Gulf, sometimes the winds will be just right. It'll push that low oxygen water, and the crabs will flee. And they get pushed up into the super shallow water, even up onto the beaches, and that's when they have a crab jubilee, and everybody goes out and picks some, and have a big party because you got easy picking crabs right up in the shallow water, right? Okay. So we talked about a couple of different types of. We talked about different types of responses or adaptations, right? They might change their physical being to change the surface area in order to get more oxygen when it's not as available. They can change some of their enzymes, they can move and things like that, but we would expect that these types of adaptations might be different depending on what kind of condition these animals live in. So this critter lives in an area that has chronic low oxygen. So the OMZs or the ODZs, these offshore low oxygen waters are chronic all year. It's really not dependent. It might vary a little bit in height and in distribution and stuff, but where they live, it's pretty much always low oxygen. And so we would expect they have things where they can reduce their metabolic rates, right? So they can do this oxyregulating behavior. And they might do things, we see lots of adjustments to the enzymes that the organisms in these types of places produce that are more efficient. They might cost a little bit to make by the animal, right? But it makes sense because oxygen is so low that they need that enhanced ability to extract oxygen from the water. Other places where we have seasonal or periodic low oxygen, like here in the Chesapeake, if you went out right now and measured the oxygen concentration, you would not likely see very low oxygen. We usually see it show up in the summer here after we have spring algae blooms that start to sink out and are respired. And so the types of things we see in a lot of these places are behavioral changes and that anaerobic metabolism that I'm not going to talk about. So any of the animals that can move are going to move out of these low oxygen areas because they're not as adapted as the ones in these places where it happens all the time. Right? Okay, so a lot of this has been kind of theoretical. So what does it actually look like? When you go out and you try to say, what's happening to these animals? What does it actually look like? So I'm going to give a few different examples here, and I'm going to talk a little bit more about adaptation as well. And so one of the things that we... Oh, can you hear me?
Afraid it wasn't working, I saw your,
Oh, no.
I saw your thumb going in.
I'm just fidgeting. So one of the things that we see with animals, and this is a little bit hard to see, but when they're raised in, this is croaker. This is from some work done in the Gulf of Mexico, but we have croaker. Whoa, sorry.
That's like the wrong thing, I was trying to enlarge it for you.
All right, now we're back.
There it is.
So this is some work from the Gulf of Mexico where they looked at how the reproductive output of animals is affected by, oh, there we go.
Does that help you?
Yeah, up at the top is right. Yeah, that's nice. Okay. How the reproductive output of animals is affected by low oxygen. And so they looked at croaker that were found in low oxygen water and croaker that were found in high oxygen water. And this is female ovaries collected from different sites. This is an example of what the ovaries look like from females collected in low oxygen sites. And this is an example of what the ovaries look like from females collected in the normoxic sites. So this is in some ways an adaptation, right? But this is a response to that low oxygen. The females their reproductive output is significantly decreased in the presence of low oxygen. Go to the next one. It's actually very similar.
That, or you want to?
No. This is just the histology. This is, if you took a slice through those and looked at it and you knew what you were looking at, they would look different. I don't know what I'm looking at. But this is what the male testes look like of that same croaker. So the animals that are raised in low oxygen have significantly decreased.
My magnifier didn't work that time. There it is.
So the males also have significantly decreased reproductive output here. So that's the ovaries again.
I was gonna say.
Yeah, I was
Yeah.
gonna say that looks the same.
This is, yeah, we don't have to zoom in, I think. This is okay. Yeah. So just as so the fish have really, they can have, hypoxia can have a significant impact on reproduction of these fishes. So not only does it, I think if you can't breathe, you're gonna die eventually. But even if you don't die, there are some real long-term impacts to the individuals and to the populations here. If you can't reproduce, it's gonna really affect your population here, right? But some fish can also acclimate to low oxygen, and in certain ways. Now, this is a study that was done on red drum. I just found this actually this morning when I was looking for another example of this. And so this is just looking at metabolic rates. We don't know anything about the reproductive rate of these fishes, but what this study did was they took a whole bunch of red drum, which are showing up more and more here in Chesapeake and are really fun to catch if you're a fisherman. And they split a bunch of these red drum into two different groups. One control group where they just raised them in perfectly high oxygen, and another group where they raised them in low oxygen, not super low, but low oxygen. Okay, and then they took both groups and they measured them under measured the respiration and a couple of other metabolic indices under low oxygen. And what they found was this. This is the amount of oxygen here on the x-axis, more oxygen on the right, less oxygen on the left. And this is a curve that shows the respiration rates. The control group is here. So this is respiration in low oxygen. The group that was raised in high oxygen has a lower respiration rate than the group that was acclimated to the low oxygen, okay? So even within the same group of individuals, within like one generation, they can start to adjust. So there's a little bit of metabolic plasticity, we would say, right? There's things that the organisms can do that they can adjust to their conditions so that they can make a living under low oxygen under certain cases.
Why are the red drums showing up now in the Chesapeake?
I think a lot of that has to do with warmer summers and things. They've always shown up here, or certainly for as long as I've been around. And I think they're migrating a little bit further north because the conditions are
They're
such.
good fishing fish?
Yeah. Oh yeah.
Never.
They're fun. They're and you can get they big ones, but they also call it puppy drum. They're small ones that like they just kind of boil on the surface. They get in these feeding frenzies. They're a lot of fun.
Kate Paul made a good living cooking them.
Yes.
Kate Paul at restaurants in New Orleans, you know, that's
Oh, yeah.
redfish, red drum, yeah.
Mhm.
Oh, yeah.
This is making me think of like, the indigenous people that live high in the Andes,
Really good. That's a good analogy.
they're used to it and when you go up there and have a really hard time. Okay.
Right, and so this is just right, and this is just one generation. Well, actually, it's pretty similar because it's thin air, right?
Right,
So
I'm
it's
just
thin
thinking they're
oxygen.
lower, higher, that's what I'm
Yeah.
trying to reverse, yeah.
This is just one generation, and so it's likely that
When you
if
say
you're
it's one generation,
so this is like they grabbed a bunch of fish, they raised them as youngsters in low oxygen, and then they tested them, right? So we don't know.
it's all done with the same
Yeah.
generation.
Yes, it's all done with the same generation.
Okay.
So this, it's very expensive, it's very time consuming to do especially with fish that live many years, right? Not a lot of federal agencies are going to give us a lot of money just to raise fish for a couple of years. So it's really hard to do multi-generational studies on these things. Some places have done it. There was a ton of work done on. Well, that's a whole other story. Anyway. That'll be for my third or fourth class. Okay. All right. So this is talking about how fish adapt, but it turns out that zooplankton can also adapt to low oxygen conditions. And this was a study done by Mary Beth Decker and a bunch of other folks. Actually, on the other side of the Chesapeake. Were you a part of this, Mike? This is Denise and.
They never asked me.
Yeah. So in this study, they took the same species of copepod, okay, which I'm going to talk about in a couple minutes, and they raised them. So they took the same species of copepod, one from Florida and one from the Chesapeake Bay. The one in Chesapeake Bay has experienced low oxygen repeatedly every summer. The one in Florida has never experienced low oxygen. And they put them in a jar. They put them in a big container that had low oxygen on the bottom, and they waited to see what happened. And the ones that had never experienced low oxygen would swim into it. They were kind of naive. They would swim into it, and they would die, or they would have decreased reproduction. They would have all these other problems. The ones from Chesapeake Bay would not necessarily go into, they would stay away from the low oxygen. They had kind of learned that was a bad place, and they don't want to be there. So they actually, the different populations raised that were in situ naturally and under different conditions, had really different responses to the presence of the low oxygen. If they knew it was there, they stayed away from it. If they didn't know,
They just
they went.
had to do it.
Step. Okay, just a couple of things about jellyfish. So jellyfish, actually, jellyfish have kind of a weird response to hypoxia, to low oxygen, and these are different oxygen treatments here, and this is how many jellyfish settle. So settling is the response of some of the larval, so I don't know, the sea nettles and lots of other jellyfish like them, they have a biphasic life cycle. So they have this, they live in the ocean, and then they produce offspring that need a place to settle, and they love oyster shells and things, so they like to settle down on oyster shells. Well, it turns out under low oxygen, you have lots more of them settle. So when the oxygen goes low, a lot more of those larvae find a place to attach, and they attach. But when oxygen is low, more of those attached larvae also die. So more of them attach, but more of them die. So there's a trigger with the oxygen that causes them to go decide that they need to settle, they need to attach, but more of the ones that do actually don't necessarily make it.
Question, I mean, I've always been told that jellyfish are in warm water. Is that true? Because in the summer you see so many here. But
So
I see them in the winter.
Yes, so there's different species. So there are different species, and there are plenty of them in the Arctic and in the Antarctic. So around here, the local species, the moon jellies are usually around in the winter. It's a different species than the bay nettles, which we see in the summer. And then we also have the ctenophores. So they're the ones that have
All those things.
yeah, the ctenophores don't sting, but they're voracious predators on lots of other zooplankton, and they show up usually a little bit earlier in the year. And there's actually two species of them. There's a wintertime one, and there's a springtime one. Yeah. So jellyfish are a really broad group, really diverse group. And so here, yes, the bay nettles prefer warm and salty, generally speaking. But there's species all around the world that prefer different things. Okay, I'm going to talk a little bit more about copepods and see if this works. All right, so I keep mentioning copepods. I may have shown this to some of you if you were at my talk last year, but this is the copepod Acarcia tonsa. It's probably the most abundant animal in the bay. Okay, multicellular animal in the bay. Copepods are probably the most abundant animal on Earth. Pretty much everywhere you find water, you are going to find copepods. These guys don't grow up to be anything else. This is what their life is. I want to see if I can.
There.
And how?
This is about a millimeter. Thanks. Yeah, that's about 1 millimeter long. Okay, and these are the biggest ones you see in here are adults. Okay. This particular species is found all over the world in coastal systems and in estuarine systems. We have lots of it here in the bay. It's been very well studied because it's found in so many places, because it can live at room temperature and things like that. So this is work that Mike had done. Copepods, particularly Acarcia tonsa, can respond to hypoxia in lots of different ways. Generally speaking, lower survival. If you raise them in low oxygen, they don't survive as well. This is survival of two different species. This is Acarcia, this is another smaller copepod here. You see when oxygen gets below 2, the survival rate starts to go way down. So they just don't make it, right? They have lower egg hatching success. So when they produce eggs, fewer of those eggs hatch when you have low oxygen. Again, this is the number of nuclei, that's the early larval stages, and this is oxygen here on the x-axis, and you can see that once you get below 2, the number that hatch goes way down. If you put the eggs in low oxygen water for a long period of time, the number that hatch goes down. So the longer they're in that low oxygen water, the fewer of them are going to hatch. Okay? And they have a reduced escape response. So it's harder for them to get away from predators. So this is oxygen. This is the jump frequency, how fast they jump. So copepods generally have, let's see if we can get that going again. So they have kind of two behaviors. They have this, you'll see some of them just sort of disappear. That's a big jump response. They can move like 10 body lengths in like milliseconds. But most of the time they're just swimming around and they're swimming around and filtering the water and feeding. So when the oxygen gets lower, their jump response decreases rapidly. Okay? So these are all just some of the things that we see. We've also done a bit of work on the connection between low oxygen and temperature, and I'm gonna skip that. And other folks have done this as well. I put this slide in because it's not a copepod, and I love copepods, but I wanted to show you that other things exist in the planet. Hypoxic, so this is an amphipod called Phronema. This is an open ocean amphipod, and this was a study where they looked at metabolism rates under low temperature and high temperature, and under low oxygen and high oxygen. Okay? Phronema is also one of my favorites. If you Google Phronema, there's a great video that someone has put together about this particular amphipod because it has a really neat life history. It actually, as a larvae, it will attack a type of jellyfish called a salp. So these are open ocean organisms. It'll attack this jellyfish like a salp. It'll burrow in and parasitize the salp. It will eat the salp from the inside out and use the body of the salp as a house.
Oh my God.
And when Ridley Scott, this is, I like my plankton stories, so you're going to be subject. When Ridley Scott was making the movie Alien with Sigourney Weaver, he actually went to a couple of different collections of animals to like get inspiration, and this is the inspiration for the alien in the movie Aliens. And so it's a parasite, not unlike the alien in the movie Aliens. So, but that's an aside. The Oscars were 2 nights ago, so I figured that was a good connection, right? But the thing that's interesting here is that low oxygen has a bigger effect at higher temperatures. Okay, so this is metabolic rate. This is 10 degrees. Okay, this is fully saturated oxygen, and this is hypoxia. So at 10 degrees under hypoxic conditions, total metabolism is lower, but it's a proportion lower. This is total metabolism at 20 degrees Celsius. Okay, so this is about room temperature, and this is what total metabolism looks like under hypoxia. So as temperature goes up, hypoxia has a bigger proportional effect on the total metabolism. Okay? And this thing's really cool. So Mike had mentioned, Mike talked a bit about temperature and oxygen, and so I found this slide, and I wanted to just go through it briefly because it talks about how temperature affects and hypoxia effects sort of work together. These are the synergistic effects. Within the ecosystem, increased temperature can increase nutrient cycling, so it increases metabolic rates of bacteria and all kinds of things like that. It can enhance primary produce, so this is algae, can make algae grow faster, things like that. Higher temperature algae grows faster. Because it's increasing metabolic rates, it increases those decomposition rates, so that's that respiration rate. So it can actually draw, high temperature means we draw oxygen down faster, and that relates to more hypoxia. So the responses to high temperature in low oxygen from a lot of animals are lower tolerance. At high temperature, there's higher demand, right? Our respiration rates are higher, our metabolic rates are higher, we need more oxygen. So our tolerance goes down. We need more oxygen at high temperature, but we can't put as much in the water because of the solubility problem, right? That leads to physiological stress, leads to behavior and migration, and it either reduces the fitness, like we saw with the croaker, right? It reduces the reproductive fitness, or it can lead to direct mortality. We saw that with copepods, and I think if you can't breathe, you're not going to last, right?
Any question?
Yes.
I'm thinking about your copepods,
Mhm.
that if there's less oxygen, they're dying. If there's less oxygen, they're moving slower, so they're being eaten by predators faster. And this is kind of the beginning of the food chain. So this is gonna have a huge domino effect down the food web.
Yes.
Okay.
Right. And it's going to be, so this is where it gets, yes, that's true. And it's very hard to predict because the fish are also going to, right? So we know the copepods have some individual responses and some population responses. The fish have some individual responses and some population responses. So when we try to add all those things together, it gets very muddy. We know it's probably not going to be good,
Oh, right.
but
That
you know,
person-
being able to predict sort of quantitatively how bad it's going to be gets pretty, especially because, you know, I'm talking about a few fish species and I'm talking about a carcharodon, so one copepod species, but the food webs are really complex. You know, the ocean food webs are really, really complex. So there's a lot of other species out there. We happen to be studying the ones, you know, frankly, because it's hard to do work in the ocean, we study the ones that are abundant and that are easy to study in a lot of cases. With copepods, with a lot of the really small plankton, we often only study the adults because the adults are very easy to identify compared to a lot of the larvae. Larvae are really similar. And so a lot of what we know is from a relatively small number of studies compared to the fact that 70% of the Earth is covered in water, right? So we call it poking holes in the ocean, right? We go out, we dip a net, and we poke a hole in the ocean, and we study stuff from there. So there's a lot we do know, but we have to sort of recognize, you know, how big those error bars are, how big that uncertainty is. Yeah. I didn't mean to sound like we don't know anything.
No, I get what you're saying. It's a lot, but.
Yeah. Okay, so everything I've talked about so far has been mostly like lab studies and things like that. So I just want to show a little bit about what it looks like when we go out in the water. And I've talked about this already, but hypoxia can affect zooplankton and fish in different ways. There can be a direct effect. If you can't breathe, you're going to die. That's a lethal effect. Sublethal effects, slow development, reduced reproductive success, and things like that. The indirect effects of habitat reduction. So if the deep water is low oxygen, you just can't go there, right? You can't make a living in that deep water. So it can reduce the amount of habitat. And that, plus some of these other things, if your jump response is decreased, it can change the trophic interactions, mean prey-predator interactions and things like that, right? But there could potentially be some beneficial effects to some of the organisms that can tolerate low oxygen in certain places. So if you can tolerate low oxygen, you might be able to use low oxygen. If you're more tolerant than your predators, you might have a place to hide. Because in the open ocean, there's nowhere to hide. There's no trees to climb up, right? If you've got a tiger chasing you or something like that, there's no rocks to climb under. In the middle of the open ocean, being a plankton, there's really nowhere to hide. So hypoxia can give you a bit of a refuge from predators if you can tolerate it a little bit. When we talk about habitat compression, it might actually make it a little bit easier. Instead of the buffet being a mile long, you might squash all the desserts onto one little 6-foot table, and it's a lot easier to get your food, right? So we're thinking about organisms in the plankton and in the ocean that live in a fairly dilute world, right? When you look at the water, the first thing you see is not the animals, it's the water, right? So they live in a fairly dilute world, and when you compress the habitat with the same number of organisms, it's a lot easier to find your food, your prey, right? And it's going to select for some species. It may not be the ones that we want, but for example, a lot of the jellyfish can be very tolerant So if you're a jellyfish, hypoxia might be the best thing to happen to you. Because it's going to make the fish go away, and there's all your food is going to be right there in front of you, right? So may not be what we would like to see, but okay. So I'm going to talk a little bit more about things that we see in the ocean, but I just wanted to show what some of the impetus for me getting involved with this when I came to work with Mike was this idea of a blue hole. And so this is the amount of zooplankton in the water. Red means more, blue means less. Okay? This is a map of Chesapeake Bay. This is from summer, I think this is 96, 95 or 96. And what we see is this big decrease in zooplankton, this big blue, Mike called it the blue hole, and it's kind of stuck with me. This big decrease in zooplankton in the middle of the bay, in the middle of summer, pretty much every year. So compared to the upper bay and the lower bay. This is much less zooplankton here in the middle of the bay. And it shows up at the same time in the same place as low oxygen. So here red means low oxygen and blue means high oxygen. This is, if you took a slice down the deepest part of the bay and then turned it sideways and looked at it, that's what we're looking at here, okay? So in the deep part of the bay, we have lots of low oxygen and we have very few zooplankton. And we went out there to look at all the different things that are happening out there in that water. And this is actually not from the Chesapeake, but I just talked about how hypoxia, low oxygen, can affect the trophic interactions. This is from a study down in the Neuse River. That's one of my favorite examples of this, because they went and measured oxygen, they measured fish, and they measured copepods. So they measured the copepods by taking nets. They measured the fish using sonar, using a fish finder on a boat. Okay, and they measured the oxygen using some kind of an instrument. So this is the red means low oxygen and blue means high oxygen. The width of the bars tell you how many. These gray bars tell you how many copepods are there at different layers in the water. And we're going to just look at that for a minute. So you can see this is a time of year when there's lots of low oxygen. This is a time of year when there's not very much low oxygen. Generally speaking, a lot more, the bars are bigger here. When there's more oxygen in the water, we get more copepods. When there is less oxygen in the water, we have fewer copepods, but the ones that are there are hanging out, most of them are hanging out under the low oxygen, right? They're hanging out in the low oxygen. So they're tolerant enough. It's low, but it's not so low that they can't hang out there, okay? This is what the fish finder shows, okay? So here, when you have lots of low oxygen, there's no fish in the really deep water, but they're kind of piled up at the same depth that the low oxygen is, right? The fish are kind of piled up here. And these are like average things. When there's lots of oxygen in the water, the fish are a lot more dispersed. They can roam around the whole thing. Now this was a zoomed in picture of some of those fish finder things. So this is time now on the x-axis, and this is depth here on the y-axis. This dashed line shows where that low oxygen was. And what I really like about this, these are mostly anchovies. This is a high frequency sonar system on the boat, and most of these dots are anchovies. And what I really like about this is it shows these anchovies kind of making a dive, right? They're diving down into the deep water. This is taken from this time period here. So we know there's lots of copepods hanging out just under the low oxygen water, right? And these fish, I always sort of envision them holding their nose and diving down and picking off a bunch of copepods and coming back up. So that's what we're seeing here, is that these animals are kind of adapting to this situation, right? And they're making the best of it. They can't live down there, but they can tolerate it for short periods of time, and they dive down into the water. This is about a 4-second window, so they're spending maybe 8 or 10 seconds at most down in that low oxygen water feeding.
So it's a stage where it's a little bit cooler and the buffet table is.
Exactly, right. That's exactly right. So this is what it looks like when you go out and try to measure this stuff. One example. That's another example of the same thing. When we go out, this is some work from Karen Wisher, and this is kind of a conceptual diagram of what's happening out there. So this is much deeper. Back here, whoops. So we're talking about, this is about 6 or 7 meters deep. So what's that, 30 feet at most, okay? These OMZs, this is what it looks like in the OMZs. And this is a little bit complicated, but one of the things Mike had mentioned that OMZs are expanding, these low oxygen areas are expanding, right? Well, they're expanding horizontally, but they're also expanding vertically, okay? So because of climate change and because of lots of things that are happening, these OMZs are expanding. And what used to be this surface layer, this gray is the surface layer here, is getting smaller. So that's that habitat compression. We have the same amount of organisms in a lot of cases, but they're being compressed. And a lot of these animals are migrating. A lot of the large zooplankton migrate. They come down during the day because it's dark down there and they can hide from visual predators. And they come up at night to feed because this is where all of the food is. And because they're queuing on, they've got, a lot of these zooplankton have eyes or some kind of optical sensor. They're queuing on light. They're migrating down to the same depth even as the OMZs are expanding, right? So what's happening to these organisms is, yes, there's more food up there because we're compressing the habitat, but they're spending a lot more time in low oxygen because they have to get into the dark to get away from some of these visual predators. Okay. Been talking for a while. I'm going to skip through a few things. How's everybody doing? Good? Okay, good. Nice.
I want to see some fish.
You want to see some more fish? All right, let me see if I can
I
get
think the
this.
second part of this should be a field trip down to Horn Point.
Well, we've talked about that. Well, I'm gonna start wrapping things up here, and I want to end on a couple of things. This is the diagram that Mike and I had put together to look at how these migration patterns and how this habitat compression, based on data. This is a conceptual model of that we put together for this. So under normal oxygen conditions, what we usually see is these black lines represent the migration pattern of copepods, and they come up to this near surface at night to feed. They swim around and they feed, and then during the day they go back down. There's plenty of oxygen around, but it's also nice and dark down there. When we have moderate low oxygen, it's low, but it's not real low, they still do a lot of that. They still spend a lot of time down there because they can tolerate it. But you'll notice now the fish have to stay up here, okay? The fish stay up, but there's the same amount of fish. So there's the same, and jellyfish, there's the same number of predators. But when you have really, really low oxygen, when it gets anoxic here, they're all gonna stay up, everybody's gonna stay up in the surface and gonna be jammed together. And this is what we see in lots of situations, especially here in the Chesapeake Bay. So, what does this mean? Well, I had a student, Catherine Lou Slater, who did a bunch of work on predation and looked at how predation changed with both temperature and low oxygen. And the thing that she found, so there's, in this graph, the bright green is non-predatory mortality. So this is the mortality on the copepods. So this is copepod death. Copepods dying just because of low oxygen, okay? And as you can see, so this is normoxic or full oxygen, hypoxic, and then this is cool, temperate, and warm water. So this is increasing temperature, and each set of bars is from low oxygen and high oxygen. So as you can see, with low oxygen, there's a lot more non-predatory mortality. More copepods are dead when we have low oxygen. And it gets the highest actually in the temperate situation here. I'm gonna take that data out, and I'm just gonna show you the predation mortality. The orange bars here are for juvenile anchovies, the blue is for larval anchovies, and the green is for jellyfish. So generally speaking, in sort of in fall, so moderate temperatures and hypoxic waters, we have increased anchovy predation. So when the oxygen is lower, the temperature is not real high, the anchovies go nuts eating the copepods here. Okay, and we saw that the copepods have lower jump responses, so they can't escape as well. under conditions like this. So the anchovies go really well, but the anchovies are not that tolerant of the low oxygen, okay? So when we get even more hypoxic water and really warm water, the anchovies don't like the really, really high water, we see increased jellyfish predation. So now we see the jellyfish are kind of the main predators on the copepods. So this plays with what we see in the summer, right? In the summer when you go out, there's tons and tons of jellyfish out there. They're the main thing eating the copepods. They can tolerate high temperatures, they can tolerate low oxygen, and the copepods have a reduced jump response. So this goes back to your question. This is going to really affect the food web because now the jellyfish are out competing the anchovies in this really hot, low oxygen water. And they're the ones that are going to be really mowing down the base of the food web here. Right? Any questions? Am I losing you? Yeah? I appreciate the honesty.
It's just a lot.
It is a lot. It is a lot. So let's see. I think what I'm going to do, I had some slides about the Gulf of Mexico. So this isn't fish, but this is shrimping. I meant to pull that out, sorry. So this was an interesting study, and I know Mike has talked about this in some of the talks he has. This is a map of bottom dissolved oxygen in 2009 in the Gulf of Mexico. So this is New Orleans would be up here. This is the Bird's Foot Delta. This is Texas here. It's Mississippi and Alabama and the edge of Florida right there. The red numbers show the lowest oxygen. So the lowest low oxygen is sort of the mouth of the Chapalaya River and the Mississippi River here, right around the Bird's Foot Delta. These down here are maps of the shrimping effort. So this is how much effort the shrimpers put, like where they put most of their effort in fishing. And it's kind of hard to see, but what we end up seeing is that the highest effort usually maps right along the edge of the low oxygen. So the shrimpers are actually kind of right on that edge of low oxygen. And there's a lot of reasons for that. The low oxygen, right? The reason we have low oxygen is because we have high productivity. We have lots of algae, right? So there's a lot of material in that, and it sinks down and it gets remineralized. But it means there's a lot of food available for things that are feeding low on the food web, like shrimp. And so at the edge of that, where there's enough oxygen, but where they're near the productivity, is an area where they seem to be happiest. And so a lot of it, I remember Mike saying that if you took a satellite view during shrimp season, you could almost map the low oxygen in the Gulf of Mexico by just mapping where the boats are, because they're always right on the edge of the low oxygen in the Gulf of Mexico. This is all about how close things are to low oxygen. So in addition to temperature, ecosystem level effects. So this is from a modeling study that uses a lot of data, again, from the Gulf of Mexico. Each of these down here is a different species. And each of the different colored bars represent different situations in the model. The darker bars are when we added nutrients to the model. The medium colored bars are when we added nutrients and we included the folks who did this, included hypoxia in the model. And the white bars are when we didn't force it with any of those ways, any of those things. And the important thing to take away from this is that there's a lot of similarity between the nutrient enrichment and the hypoxia situation. So excess nutrients are going to cause the food web. These are all different members of the food web, all different types of things. Nutrient enrichment and hypoxia have very similar effects on the animals that are in the food web. Okay? So by reducing hypoxia and reducing nutrients, we can have we have to do both at the same time in order to affect changes in the food web, right? So they have synergistic effects, both of them together. Adding nutrients and increasing the amount of hypoxia have very similar effects on most of these organisms. Question? Yes?
on that slide, what's the difference between the P and the C of Menhaden, for example?
So this is biomass and this is catch. Okay, so this would be Menhaden catch, and this is Menhaden biomass. Does that make sense?
Yes.
That's why we don't spend a lot of time catching jellyfish, but there's jellyfish biomass in the model. So this model also includes fishing pressure and things like that. So this is the kind of thing that, this isn't necessarily an operational model, but I think this is Kim de Mutzer. Yeah, this is Kim de Mutzer. So she is a colleague Mike and I have worked with over the years, and she does these food web models, and they look at different scenarios about either restoration practices or changing fishing practices and things like that, and then examine how that is predicted to change the food web based on all the information that we have on different members of the food web. You'll notice she doesn't have zooplankton in here, which is a point of contention that we have raised.
I don't know why your pointer's not.
Maybe battery thing.
Oh, I can see it on your hand.
Yeah, it's good up there. Okay, so the displacement of fish, like how fish are pushed in situations where we have hypoxia varies on a whole bunch of different things. The severity of the hypoxia, the predator-prey interactions, other physical factors that affect the biology, like the temperature and salinity, the tolerance of the animals to those things. But generally, fish have better survival when there's more oxygen. That's not very surprising. And some species we know can use moderate to low dissolved oxygen as a refuge, but some cannot. Some have to get away. Some of the big ones that have big oxygen demands are going to have to move. Whereas some of the ones that don't have as high an oxygen demand can actually live there and kind of hide. So this was from a paper that was written by a colleague who was at Horn Point, is now out west, and it's pretty much my last slide. And so this is sort of a conceptual diagram of winners and losers under different conditions. This is based on the Chesapeake Bay food web here, and it's just to highlight the fact that hypoxia can really restructure the whole food web, and it can push the food web into these long-term changes, sort of a new stable state. So we have consistent hypoxia in the summer around here, and we've kind of switched. This is the old food web. It shouldn't be on the right. These should be reversed. But right now, well, I guess if this is the one where we want to get to, right now what we have is we've got higher temperatures and we have lower oxygen. We've got lots of, you see the jellyfish is bigger over here. The sea nettle is bigger. The ctenophores are a little bit smaller. We have a fair number of anchovies. They seem to be doing okay. But copepod numbers are actually pretty much down over the last 50 years on average. We don't have a lot of monitoring for zooplankton, but from what we can see, it's decreased. And we have a lot of the zooplankton going into the jellyfish and the ctenophores and things like that. When we move to a different stable state, we have different predators that are going to have a bigger impact on different parts of the food web. So these changes can flip-flop depending on how we treat the system, depending on how we affect nutrients going into the system, which are going to affect the hypoxia, depending on climate change and things like that. But there's always going to be winners and losers. There's always going to be some animals that are going to do fine, and there's always going to be some animals that are going to be more impacted than others. And I think that's pretty much all I had for today. That's from one of our cruises where we studied hypoxia. That's just what one of our sample jars looks like when it comes up. These are all copepods, pretty much all copepods, probably 90% copepods. Thank you.
Thank you. Sounds like a cereal.
Yeah.
Let's say in the atmosphere,
Yeah.
the gases diffuse very quickly, typically. You know, if you had low oxygen
Yeah.
and higher oxygen, they equalize very quickly.
Yep.
So how come there isn't any of that going on underwater?
Well, do you remember Mike's slide with a particular building in New York City that was about 300 meters? So the diffusion rate, you had a picture of Trump Tower, and then, and next to a copepod, and a copepod's about 1 millimeter. Trump Tower is 200 and something meters high, right?
Right.
That represents the difference in diffusion rate. So diffusion rate in air is about 300,000 times faster than the diffusion rate in water. So that's why diffusion into the deep ocean is not really a feasible way to get oxygen in. But at really small scales, across a membrane, it works.
It's just, so it's the physics of
It's the
it
physics of it, yes.
to get through the liquid
Yes.
versus
Yep.
gas to gas.
Yes. Yep, that's exactly right.
Because the particles are moving slower in the liquid than they are in the gas.
So to wrap up between the two classes, when we talk about rising temperatures,
Mhm.
where does that leave us?
Well, just get, as Jamie showed, the warmer the water, is, you know, and coastal waters can warm up faster than the open ocean because they're shallow. The water in Chesapeake Bay hangs around for about 3 months before it flushes. Same thing with Long Island Sound. So these coastal bodies where we live are heating up faster. When they heat up, they hold less oxygen, right? And then the animal respiration, it's not like us. Our respiration is independent of temperature, but theirs is directly proportional to temperature. So for the little bacteria, protozoa, as it warms up, they're using up the oxygen. So it's a double whammy.
So they're
You know,
dying more to
the
the
water
bottom.
holds less and they're losing more to respiration. So poor copepods and fish, they're having a tough time getting enough oxygen to begin with, are really getting affected.
So we
It's
need
something
a
they
big
don't
pump,
appreciate with climate change.
we need a big pump at the mouth of the Chesapeake that's just constantly moving over
So-
water.
So they've actually, there are folks, in fact, there's a group from the Chesapeake Biological Lab, one of our sister labs, part of UNC's, who actually did this in a small tributary up outside of Annapolis. They were having some water quality issues, including hypoxia and deep water, and they put a pump. So they put a pump in, and they were like, oh, well, this should probably fix it. And it didn't. And it didn't for probably a variety of reasons. They just did this a couple of years ago, so they're still working through a lot of the data and the synthesis, but these are really complex systems. And, you know, the water is moving, the animals are doing different things. When you start changing the oxygen, you're gonna change the organisms that are making a living there. So these things are there's a lot of, we call them nonlinear interactions, right? Sort of unintended consequences, because not everything is at the same linear scale. It's not, oh, if we do this, it'll get better, and this will increase this way. Some things go like this, some things go like this. You know, there's all kinds of different responses.
Doesn't work.
And when you add them all up, it doesn't necessarily, you know, there's a lot of unintended consequences. But people have talked about putting things in the water to either pump oxygen down or make other
Well, you can
changes.
do that in a small, like those little ponds we have over in Eastern Club East, you can
Yes.
do that sort of thing there.
That's right.
Yeah.
Small scale, yes.
Yeah, you certainly can do it on small scales, but you know, the vastness of the ocean, I can't remember Mike's, you had an analogy for the size of the dead, the dead zone in the size of Massachusetts, for example.
Yeah, the Gulf.
Yeah, in the Gulf, yeah. The Chesapeake is a little different because the Chesapeake is the drowned river valley of the Susquehanna. So the Chesapeake's dead zone is kind of long and narrow. So it's deep. It can encompass like up to 60% of the water in the deep part. But because the Chesapeake has this hypsograph, so you took a cross section of the Chesapeake, it's very, very shallow, very, very shallow, and then dips down. And I think we said last week, you know, it's 3 or 4 meters deep. I think is, you know, under 10 feet is the average, or around 10 feet, I'd say, is the average depth of the Chesapeake. But so all the hypoxia is confined to that deep area where you don't have penetration from the surface, where the wind doesn't really reach in most cases, and so the area that's hypoxic in the Chesapeake is much deeper in general than a place like the Gulf of Mexico, where it's only like a meter deep, but it covers a huge area.
So how does this impact our, the watermen and the production of oysters or crabs or fish?
So like, for example, in the Gulf, in the Chesapeake is a little bit different because of how the hypoxia, that's, there's not gonna be much alive worth fishing for, I should say,
When that's.
in the low oxygen water, right? But in the Chesapeake, that's confined to this narrow, long place in the middle of the bay. So, if you think about from where the ports are, but if you think about a place like the Baltic Sea, where they've had chronically low oxygen for probably 100 years now or so, or the Gulf of Mexico, where you have bottom water, the hypoxia is always on the bottom, right? Because unless you get a big wind event, it's always on the bottom because we're putting oxygen in at the surface. And so if you are looking to fish for things that live on the bottom and those things have to move, you have to go much further to find them. If all that, if you're used to fishing an area of the Gulf of Mexico and now it's suddenly low oxygen, you gotta get to the other side of it, for example. That means you gotta travel a lot further for the same amount or maybe even less of your fish. So it has huge economic impacts.
The 826 years was, as I understood, the time that it would take estimated for a block of
Yes.
water from one place to go all around the world.
Yep.
But in large basins like the Pacific,
Mhm.
like the Indian Ocean, isn't there a lot shorter cycle of exchange?
So this is like, that's yes. And so when we think about like a block of water in my physical oceanography class, we call it a parcel of water, right? So this is like the longest that some of this, so when we think about, if you hear about carbon sequestration or carbon dioxide removal, and they talk about putting carbon dioxide into the water, into the deep water, this is what they're talking about, is putting it in those places. Now, in other places in the ocean, yes, the residence time is much shorter. There's places where where you have a lot more exchange between certain deep water areas and shallow water areas. And there are, if you remember,
Oh,
that's right, don't worry about it.
I took it down.
That's okay. There's places where the water will come up. So this is sort of on average. This is an on average, the water that is the deep water that is formed in the North Atlantic takes about 800 some odd years or more to come up in the Pacific.
So if you put a bottle in the water with your little nose, it's going to take a while.
If it was neutrally buoyant, yeah, for that type of water, yeah. So, but yes, in like local systems and in regional systems, it's gonna be a lot shorter. Which is one reason why I think a lot of oceanographers have raised concerns about using the deep ocean as a reservoir for, say, carbon sequestration and things, because this, that 800 is an average number, and you're gonna have, it's likely, we've been putting carbon dioxide into the atmosphere for 200 years or so, right? Since the beginning of the Industrial Revolution. We're starting to see elevated carbon dioxide concentrations in places where it's coming up already, right? So it's not necessarily a long-term solution. On average, it takes about 800 years, but in 800 years, we still got to deal with it.
Well, if we're even here then.
Well, somebody's got, our ancestors will have to deal with it, right? So somebody will have to deal with it. If I'm not going there. I'm not ending on that dark note.
That's a robust framework of the world.
Well, so are you saying that it would make more sense to do the carbon sequestration in warmer waters? Or in smaller?
All right, so this is my opinion. It makes more sense to figure out a way to stop putting carbon
That's what I
dioxide
was
into the
gonna say.
atmosphere. Yeah, I mean, yes, it would be great if we could pull some out, but I don't think putting it into the ocean is a long-term solution. We could figure out a different way to sequester it, fine. But
if you sequester it into deep, cold water, then it's not gonna come around for a long time. So then maybe it does make sense.
it's
More sense than
putting
putting it in
it
warm.
putting it off,
Yeah.
so.
like
Treating the
taking
symptom
hand
and
down.
not the disease.
Yes.
You might have a question from the,
Oh, sure.
William?
Larry, Bill, Robert, any questions?
Don't have anything, Jim.
Bill's saying he's
Well,
good.
thank you for tuning in, though.
Yeah. Well, thank you so much. I really appreciate your doing this. for
Sure.
us.
Thank you for taking me. I know it gets weird, but thanks for tuning in.